Welcome to Parasitic Diseases. Today we're going to discuss the malarias, and that includes Plasmodium vivax, Plasmodium ovale, Plasmodium malariae, Plasmodium falciparum, and Plasmodium knowlesi. And as we'll see, these organisms together represent the largest threat to human life with regards to infectious diseases. And therefore, it is of paramount importance that we learn as much as we can about the biology of this organism before we discuss the clinical aspects. So to know where all of this starts, let's put malaria in context with the rest of the organisms that share a common biology. The group is called Apicomplexa. The Apicomplexa include the malarias, Cryptosporidium parvum, which we'll discuss next, and then finally Toxoplasma gondii, which, although it is not the most life-threatening organism that we'll be dealing with in these presentations, it's the most prevalent organism with regards to the infection of humans. Toxoplasma probably infects more humans and other mammals as well than any other organism on the planet. But as we'll see, there are some huge differences with regards to the way in which the clinical manifestations of these apicomplexa are seen in a human population, and the way in which the immune system deals with them as well. So the apicomplexa, as you can see diagrammatically here, starts with the apical end, of course, which is referred to as the polar ring, and it is the exit point for a number of interesting secretory functions that rely on romantically named organelles such as rope trees and micronemes. This has a very long duct, and it produces materials which aid this organism in gaining entrance into its host cells. So that's what the apical end of this organism is all about, and that's the business end of the apicomplexa. And interfering with that process, of course, would be a logical strategy if we were to, let's say, invent a monoclonal antibody which interferes with that, or perhaps a drug that interferes with some of the substances that these organisms secrete. But we're getting ahead of the story right now. Just know that malaria fits well within this apicomplexa, and as far as we know, there are no overt mechanisms for locomotion. These organisms are all intracellular, and they gain entrance from cell to cell by some very unusual mechanisms. And as we'll see, and I think everybody's probably familiar with the fact that malaria is transmitted by mosquitoes. So we've now got a two-part system, as we had for, let's say, the leishmania, for sandflies, and the trypanosomes in Africa for tsetse flies, and the trypanosomes in South America and Central America with regards to reduvid bugs. And now we've got yet a third vector species to talk about, and namely the Anopheles mosquitoes. So where do we find these parasites anyway? So they're found primarily throughout the tropical world, and these are rough estimates as we see. The colorized regions of this world map roughly expresses the location for both Plasmodium falciparum and Plasmodium malariae. They overlap in their distribution. And as we'll get to as the clinical aspects of this unfolds, it's Plasmodium falciparum that we're primarily worried about when we talk about mortality rates and morbidity rates. The rest of the malarias are fairly benign compared to Plasmodium falciparum with regards to the effects that they have on the intermediate host. And I'm saying this as a way of alerting you to the fact that we are not the definitive host for the malaria parasite. Rather, the Anopheles mosquito turns out to be the definitive host, and we are mere carriers of the stages that the malaria mosquitoes acquire by sucking blood from an infected human. This shows the distribution for Plasmodium vivax, and we have another one to add to our list, which has recently emerged as the result of environmental disturbances in these regions throughout Southeast Asia and throughout the countries of Indonesia and its island communities. It's called Plasmodium knowlesi. It's usually a primate malaria that does not infect humans. But as our population has grown and as we have encroached into areas where these primates live, we too are susceptible to those infections. And in fact, there is actually another one that has emerged in South America as well for the same reason, but it's of a minor importance. So we're not going to mention it right away. What's really important to know is, how do we even come to know about malaria? How did it evolve with regards to, let's say, the germ theory, or perhaps whether or not vectors are important for the transmission of diseases in general? So what we see here is a rogues gallery of people who are given credit for discovering various aspects of the life cycle of malaria. We have Lavrin and McCallum. Both of these people, in independent studies, discovered the sexual stage of malaria without realizing what they were looking at to begin with, by actually drawing blood from an infected patient and then just letting it stand around for a couple of hours. And when that happens, the male parasites of the male-female parasite complex for malaria, and so I'm giving you a little bit of a hint here that this is a sexual organism, has both male and females. The male organism of malaria actually produces these long, sinuous, what turned out to be sperm. And the process became known as exflagellation without knowing exactly what they were looking at. And it later on turned out, when we pieced all the life cycle parts together, this is the stage that takes place inside the stomach of the mosquito after she takes a blood meal. So how did we learn that malaria is transmitted by mosquitoes? And then we have to jump all the way over to Sir Ronald Ross, in this case just Ronald Ross, before he was knighted. Ronald Ross was working in India, and at the suggestion of Patrick Manson, decided to do some studies with regards to malaria to see whether or not he could show that it was transmitted by mosquitoes. And because his resources were limited, he was forced to work with birds. And in doing so, he eventually did work out the fact that bird malaria, and indeed birds have malaria just like we do, but different species, that bird malaria is transmitted by what later on became known as the culisine group of mosquitoes. And as hard as Ross tried to make the same connection between culisine mosquitoes and human malaria, he failed. And it was very simple as to why he failed. He failed because the culisine mosquitoes are not responsible for transmitting malaria to humans. That discovery waited until Bignami and Grassi, working in collaboration with several other people in Italy, exhausted the culisine mosquito species where they lived, trying to show the same thing for human malaria, and the only mosquitoes that appeared to be left were the Anophelines. Lo and behold, it turned out that the Anopheline mosquito was indeed the culprit for transmitting malaria from person to person. And in fact, the Anopheline mosquitoes are not responsible for transmitting malaria to birds. So here we have two different groups of mosquitoes, two different groups of malaria, and of course, two different groups of investigators. A lot of controversy arose as the result of the discovery by the Italian group that indeed human malaria is transmitted by a different kind of mosquito than the one Ross discovered in India. And that controversy was unresolved. And in fact, it even got exacerbated by the fact that eventually the Nobel Prize Committee awarded Ross the Nobel Prize for discovering that malaria is transmitted by mosquitoes, rather than awarding it to Grassi, who really discovered all of the essentials for the malaria mosquito transmission to humans. That controversy remains in place today, and I'm not going to spend too much more time on it, except to say that there are still the fans of Sir Ronald Ross as the discoverer of malaria in mosquitoes, and then there are the other group that favors awarding more credit to the Italian group. We won't pursue that any further because actually we would like to get on with telling you about malaria in general. It turns out, because the Earth is now populated with some 7.4 billion humans, about half of those people live within a risk area for acquiring malaria. That's an enormous number of people. And annually, this is a rather interesting way of expressing the data because, as you'll notice, the risk is listed as billions of people. And each one of these districts is the Southeast Asia area, African area, European, and we have Central and South America as well, to show you the distribution of malaria just from falciparum risk here in this column, and of course Vivax in this column here. The risk is huge. And so therefore we have a lot of transmission that goes on throughout the world. We have a lot of morbidity, and we have a lot of mortality. But considering how many people actually get infected with malaria every year, and those are just rough estimates, let's say it's upwards of 200 million to 500 million people. The exact numbers can never be known. We certainly could keep track of how many people actually die from malaria. And the death rate is much, 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 much lower than the infection rate or the morbidity rate, which says that the mortality rate for malaria is very low. However, because the numbers are extremely high for infection, the numbers of deaths are also high. And most of those deaths occur in Africa. They occur in sub-Saharan Africa, and they occur mostly in children. So every year, Africa takes the hit for malaria deaths with regards to worldwide death rates. Throughout the world, each year, approximately 500,000 to 1 million people die from malaria, despite our best efforts to control this infection. And so it remains a huge, huge problem indeed. If we were to express the presence or absence of malaria based on the number of people that acquire the infection, and then express that in terms of the size of the map, how you would expand or reduce the size based on the prevalence of Plasmodium falciparum, in this case, in the blue map, you see that India and Southeast Asia are out of proportion. Africa is somewhat shrunk down in some places and enlarged in others. And of course, the North American continent has no malaria whatsoever that's indigenous. And the South America also, with regards to at least Plasmodium falciparum, is a rather small profile, and you can say the same thing for Plasmodium vivax. Those two are overlapping. Here's the real essence of all of this, and it says 3 million deaths a year, 1 million in Africa alone. Those numbers vary greatly with regards to weather and climate. And of course, malaria is transmitted by a waterborne insect. Malaria breeds in water. That is to say, the malaria-carrying mosquitoes breed in water. So if it's rainy, you get more malaria. And if it's less rainy, of course, you get less malaria. That's shown well here because the watersheds of Africa, shown in this map, with all the drainage systems from the Congo and all the West African river systems, people locate along those river systems because of the access of water and travel. And when it rains and those rivers swell up and the swamplands associated with them also acquire water, the malaria mosquitoes go crazy. They absolutely reproduce because that's their time. And in order to finish their reproductive cycle, of course, they have to suck blood in order to lay the eggs. The mosquitoes are interesting in that way because only the female mosquitoes do the biting and take the blood. And they use that blood not for their own food, but rather for the protein source to make eggs with. The male mosquito, as well as the female mosquito, derives their own nutrition from plant juices. This map shows the population density of people and their relationship to the river systems of Africa. And there's a one-to-one correlation between either standing bodies of water like Lake Victoria and Lake Tanganyika, as you see over in East Africa, or in West Africa, all the river systems there. And that's the basis for the epidemiology for malaria throughout Africa. We could go on about these statistics. We'll come back to them when we talk about control measures and ways of preventing malaria. Here's a wonderful shot of an up-close and personal Anopheline mosquito. Her proboscis is buried into the thumb of one of the authors of parasitic diseases, Robert Gwadz. This is an uninfected mosquito, of course, raised in the laboratory, but he sacrificed his own blood supply to allow us to see up close and personal exactly what this mosquito is experiencing when she lands and takes her antennae and her proboscis. And this is here are the antennae over here, and the proboscis is over here, and the business end of the proboscis is now revealed, and it penetrates into the skin, into a capillary, and eventually she fills up with blood. And you'll notice that she has no use whatsoever for the plasma portion of this. So in terms of blood banking, Anopheline mosquitoes were the first to perform plasmapheresis, of course. We may have even gotten that idea from watching them feed. But they want all the formed elements of blood in order to convert that protein source into their eggs. If we look at the way the malaria parasites appear on smears of blood from the intermediate host, we get a picture that they're all basically similar. They go through similar stages to reproduce inside the red cells, and we'll get to that when we talk about the human aspect of the infection. And but there are major differences as well that a trained technician can tell you whether or not you're infected with Plasmodium vivax or Plasmodium falciparum or any of the other malarias for that matter. And we now have molecular testing systems that will also reveal the differences of these species. And here are the other two, just to show by comparison. This is part of that same set of exquisite watercolor paintings that were part of a set. And now we have to get that same artist to render Plasmodium knowlesi to make it a same scale so that we can actually see up close what these all look like together. Malaria is not without attention from the research community. In fact, neither are we. So, in fact, we have now completely sequenced the human genome. We've completely sequenced the malarial parasite genome, and in this case, it's Plasmodium falciparum that we've sequenced. And we've sequenced Anopheline stevensi, the prime vector for malaria in many parts of the world. So we now have the three-part system all together, and you would assume then that because we know everything about the genes, that we should be able to tell something about the way these things interact together, but we're still busy deciphering how the genes play out in terms of our own development, and then certainly we're behind the eight ball, I would say, if I had to put it any other way, of describing how the genes of malarial parasites are active in various parts of both the human and the mosquito hosts. But work is going on all the time to try to reveal these relationships, and as they become available, of course, we will make updates to parasitic diseases to include those data as well. So let's start with an overview of the definitive host, namely the mosquito. The mosquito, as I mentioned, is a female Anopheline mosquito. She flies and detects the presence of a human based on our secretion of things. And one of those things is carbon dioxide, of course. Carbon dioxide forms a gradient away from our oral cavity. And female mosquitoes are very good at picking that gradient up. And there's an organ called Johnson's organ, and it's located right at the base of the antenna, as shown schematically right about here. And that organ has the ability to detect volatile substances given off by its host species, in this case, us. So we not only secrete carbon dioxide, but other long-chain carbon compounds as well, volatile compounds that form very, very small but distinct gradients. And this mosquito species can hone in on those very well. Once she detects us and gets close, then she picks up on our heat signal. 37 degrees, right. Now I've got the right species. The next thing she does is land. She carefully extends the proboscis and penetrates the skin. And what she's really looking for is a capillary, right about in this area, right over here. And she finds it remarkably well because the sensitivity of heat varies. Of course, this is the dermis. And she can actually she has a heat-sensing proboscis. And once inside the capillary, the flow of blood begins. She begins to fill up her diverticula, which stem off the stomach, and she packs it filled with red cells. And of course, most of the red cells that she ingests, are not infected. If this is a low infection, she has to ingest a lot of red cells in order to acquire both the male and the female cells in order for this cycle to start in her. But that does happen in endemic areas, of course, because people are infected with malaria, and they last rather a long time. It can last up to months in some people, so that mosquitoes have access to these infected individuals for a long time during the course of a year. So in come the red cells. Most of them are digested. When the infected red cells encounter the stomach and the digestive juices, McCallum and Laveran's observations become obvious. The male gametocyte, as it's called, produces sperm, or the equivalent thereof, and the female or gametocyte remains unchanged. And of course, it's minus the infected part of the red cell, which they are covered with. So they're now free to interact. And so the sperm fertilizes the egg, so to speak, making it into a zygote. The zygote is called an ookinet. And the ookinet then penetrates between the wall of the stomach of the mosquito. Doesn't actually go into the cell. It actually penetrates between these columnar cells in the stomach and comes to rest underneath the membrane that holds all those cells together, and begins its development towards producing a stage of the infection that the mosquito can then transfer to the human host. This stage is called the oocyst. So the ookinet becomes the oocyst, and the reproductive cycle from this point on produces haploid organisms. So it's a diploid organism here. It's a diploid organism here. But once it transforms into the oocyst, it begins to produce, by a reduction and then division process, the next stage in the infection, which is known as the sporozoite stage. And the sporozoite is the infectious stage for humans. These leave the oocyst when the oocyst becomes full, and they actually circulate throughout the open circulation of the mosquito. The mosquito doesn't have any capillaries or arteries or arterioles like we do. The mosquito has a heart, and it's a 3 or 4 chambered heart that pumps fluid throughout the entire surface of its interior cavity. And the fluid that it pumps is called hemolymph. And the hemolymph bathes all the organs, and that's how they acquire their nutrition. The hemolymph eventually circulates up into the head of the mosquito and down into the salivary glands, and that's shown here. And when that occurs, the sporozoites then, they sense the fact that they're in a different place. There is a recognition of the fact that they are near the salivary glands. Perhaps there's a receptor involved. Perhaps the metabolism of the salivary gland cells themselves give off a signal that tells the sporozoites, if you penetrate these cells and then get into the duct of the salivary gland, then the mosquito can then transfer them into the next host. And that's how that actually works. This whole process takes about a week and a half. I should also tell you that mosquitoes don't live more than 2 to 3 weeks. So these mosquitoes are in a big hurry in order to complete the life cycle, not of the parasite, of course, but of their own life cycle. So it takes them about 3 days to process the proteins inside their diverticula into their egg-producing center, the ovaries, of course. And the vitelline material is then transformed into eggs. The eggs are then laid on water, fresh water, not salt water. And that takes about a week and a half to complete. That mosquito can then go back and bite another individual. And in fact, in order for malaria to succeed, the mosquitoes that are infected actually have to bite twice. The first time to acquire the infection, and the next time to transmit the infection to another host. Again, just for references, here's that same depiction, real life, of the mosquito completing this manifest destiny for the Anopheles mosquitoes and for the parasites. Here's just an up-close view of an exflagellation event. Here's what the oocysts look like on the wall of the stomach of a heavily infected mosquito. This is a very heavily infected mosquito indeed. Here we see the sporozoites with its nucleus and cytoplasm on both sides. Remember, these are haploid organisms, and these are free in the hemolymph, but they accumulate in the salivary glands. One of them is shown here. They're rather nondescript. And then finally, the mosquito injects them into the host to begin the life cycle in the mammalian host. So let's talk about that now. The sporozoites are injected as the mosquito is injecting all of her pharmacopoeia, so to speak, of salivary gland secretion proteins. Some of them cause vasodilation. Others cause the area to become anergic so that you don't detect the fact that the mosquito is actually penetrating your skin. And others allow the ease of blood to transfer through that very narrow stylus, so there are some anticoagulants involved as well. The sporozoites go in with those secretions, and they get into the general circulation. And indeed, there is a receptor for the sporozoite in the liver, and in no other organ does that occur. And the parasite then goes to the liver and penetrates and gets involved in a parenchymal cell. So each sporozoite has the capacity for infecting the parenchymal cells of the liver. And when it does, it begins another cycle of reproduction, still as a haploid organism. And these little dots that you see depicted here, and this large structure that you see here, is called a cryptozoite, and the cryptozoites are inside this large cyst that's created inside of each of these parenchymal cells. And they produce thousands of cryptozoites. So many so that eventually the cell is stretched to its limits and it bursts. When that occurs, the organisms gain access to the liver's form of the circulation. Circulating cryptozoites are now free in the blood, and they are distributed throughout the body. And in doing so, they encounter lots of red cells. And very often, students will ask, well, can these cryptozoites re-enter a parenchymal cell and continue the cycle inside the liver? And the answer is no. Once they're released into the blood, they can never reinfect or never infect the liver. In fact, they are not the stage that infects the liver. We now know that it's only the sporozoite that's capable of carrying out this first step. Now, as they circulate and encounter red cells, an entirely new set of conditions exist, and the merozoite, now it's known as a merozoite. So the cryptozoite and the merozoite are the exact same form of malarial parasites. Only one is inside of this cyst, and the other one has been freed. So once freed, they're now referred to as merozoites. They infect red cells. Each red cell, let's say, is infected with a single organism, and it begins to look like this. It's a little ring with a double dot. The double dot represents the nucleus. And from this point on, the organism then begins to multiply within each of these red cells. And this gives some hint as to the clinical consequences of exposure to malaria, because basically it's an infection of red cells, and if enough red cells are infected and consumed by the parasite, then the host begins to suffer, because obviously we need our red cells for lots of things, right? Not the least of which is to carry oxygen to our cells and to take CO2 away from cells and put it back into the lungs. But other things too. What's shown here, and we're showing Plasmodium falciparum first because this parasite creates the most pathology and the most damage, and therefore the host itself suffers greatly from this. And in fact, this is one of the lethal forms of malaria. It's shown to reproduce in a very unusual place. It doesn't reproduce within the circulation of the host. It reproduces after a very interesting phenomenon occurs, which results in little projections from the surface of the red cell during the reproductive phase of this organism. It's a histidine-rich protein that the parasite produces that is included underneath the membrane of the red cell, and it makes the red cell sticky. And this sticky red cell, when it encounters a precapillary arteriole, then sticks to the endothelial cells via this histidine-rich protein. It slows down the flow of blood through the precapillary arteriole, and it reduces the oxygen tension, which is critical for the reproduction of this parasite, because this parasite turns out to be a microaerophilic parasite. It requires a greatly reduced oxygen environment in which to reproduce. And so for many years, investigators in the laboratory were trying to grow Plasmodium falciparum in vitro, and they were imitating, of course, the conditions that you would find in normal blood, and that's a highly oxygenated situation. And for many, many years, they were unable to get this parasite to actually reproduce inside red cells and transfer to another red cell, then reproduce in that red cell and transfer to another red cell. But eventually, two investigators, Jensen and Traeger, working at the Rockefeller University, were able to place tissue culture plates of merozoites plus uninfected red cells in a reduced oxygen environment using a very simple anaerobic chamber that we would use to grow, let's say, Clostridium bacteria in, and placing these little dishes of red cells and parasites together, drawing out the oxygen, and then coming back the next day. Imagine their excitement when they found that the parasites had not only undergone a reproductive cycle, but in 2 or 3 days later, they were able to complete their life cycle and go to the next set of red cells. And therefore, they established the in vitro cultivation of this parasite. And that led to a number of discoveries. Of course, once you could grow it in vitro and didn't need a host like an Aedes monkey in this case to serve as a experimental animal host, you could do lots of things and study its metabolism, study its the effect of drugs on the reproductive cycle, etc. This is called deep vascular schizogony. The word schizogony refers to the division of the parasite, and it's not a straightforward division cycle. It's not a binary fission cycle as we would find, let's say, for the trypanosomes or for the leishmania. In this case, they all divide, but they remain connected to the same cytoplasm. So it's like a little central unit of cytoplasm, and each one is a little stalk off of that central cytoplasm, and the organisms then mature as a interconnected syncytium of tissue. Interestingly also is the fact that what they're growing from is the consumption of the red cell's hemoglobin. That's its food, primarily. It also requires an exogenous source of ATP, which it derives indeed from the red cell as well. The malarial parasites consume the globin portion of hemoglobin and discard the heme part in a very unusual way. They accumulate large amounts of heme in the center of the cytoplasm that they're all connected to, and it forms a complex called hemozoin. And it actually crystallizes. This is a pure heme group stacked on top of each other in an ordered fashion, and the enzyme that does this used to be called the stacking enzyme because obviously that's what it was doing. It was taking free heme groups and stacking them on top of each other. We now know that its real name is heme oxygenase. It's a parasite enzyme, and it's very important for the metabolism of this organism because without being able to discard this heme group in a meaningful fashion for the parasite, the heme group becomes a toxin for the parasite. And as we'll see, this is one of the drugs actually acts by preventing this process. But let's get on with the life cycle. Eventually, of course, the organism consumes all of the raw materials for its growth. It then causes the red cell to burst, and the parasites are then released so that they can affect another red cell, and another red cell, and another red cell, and so on until such time as the host begins to sense the presence of the parasite. There's absolutely no reaction whatsoever to the cryptozoites. This occurs without any interference from the host. There are no immune responses directed against this stage at all that we know of. But once the parasite starts to interact with the red cell stage, it begins to release things into the bloodstream during its division cycles, which the immune system picks up on and begins to respond to in a way that could prevent the reinfection of more red cells if allowed to go untreated. And in fact, if a person is to survive this infection, the mechanism of survival is due to the fact that we produce an acquired immune response against the antigens associated with the merozoites, and also because we're reinfected again and again and again in an endemic area to the antigens associated with the outer coating of the sporozoites. Now, that antigen has been well characterized, and we'll come back to that when we discuss the development of vaccines. And I must tell you in advance, unfortunately, we don't have any vaccines right now that we can use, but we have explored the possibilities in a number of the stages of this parasite. For this parasite to complete its life cycle, of course, it has to be taken up by another mosquito. And in order to do that, it transforms a portion of its population from this, let's say, erythrocytic stage, which is a division cycle stage, to a non-dividing erythrocytic stage, which we have referred to earlier as the macro, this is the female, and micro, this is the male, gametocytes. And they're referred to as gametocytes because these are pre-sex cell stages that circulate freely throughout the host. And these stages are picked up by the next mosquito, and in the gut tract of that mosquito, then we can go back to the mosquito cycle and say the next thing that happens is that the digestive enzymes in the stomach of the mosquito digest away the red cell portion, releasing the free parasites. They sense the fact that they're in the lumen of the stomach of the mosquito. Exflagellation occurs. The sperm and egg unite. They produce the ookinete. The ookinete crawls underneath the cells, produces the oocyst, and it resumes the cycle again. So this is the intermediate host cycle, and this is what results from the definitive host cycle. Very complicated biology, both in the mosquito and in the mammalian host. And it's taken us a long time to decipher all of this. Just some visuals to reinforce what I've already said. Here's a scanning electron micrograph from a freeze-fractured sinusoid in a liver, showing you one sporozoite. I don't know how they got this, because it's by luck and by chance that you'd actually ever find something like this, and it's like finding a needle in a haystack as far as I can see. But lo and behold, they've found a sporozoite in the process of invading the liver by first attaching to receptors on these endothelial cells in the sinusoids, and then once they do that, they are able to then penetrate into the parenchymal cell and then start the process of developing the cryptozoite stage. And here we see an up-close and personal cryptozoite filled parenchymal cell. Each one of these little dots that you see here turns out to be a cryptozoite when it's released into the blood. Again, the terminology changes from cryptozoite to merozoite stage. And so here we see the merozoite depicted, both in reality and to translate that down into this diagram. We see the prominent apical end over here with the rope trees and the micronemes and dense granules and all kinds of other structures inside. All of these function for the parasite in enabling it to gain entrance into a red cell. And here's one in the act. This is the red cell membrane. And you can see that the parasite has caused an invagination of a portion of the red cell membrane, and it has attached itself in two separate places. And actually, this, if in 3 dimensions, this is a single place that's got dimensionality to it, and it goes completely around the parasite. And what you're really looking at is the parasite making a pocket inside of a red cell. And so, if you wanted to be anatomically correct, you would say that the parasite is not intracellular because it creates a pocket for itself and it stays there. It's known as the parasitophorous vacuole. And a more detailed view of this, a summary of all of the things that we have learned by observing various portions of the penetration process. We know that there's a lot of activity with regards to the activation of red cell membrane proteins that are floating free inside of this red cell. It has a reserve of protein that it can use, that the parasite is obviously taking advantage of, that it can make a little home for itself without altering the size of the red cell, which is quite unusual. And there are very few parasites that actually infect red blood cells, and this happens to be one of the ones. In order to identify the life cycle stages of malaria, all we have to do is to take a blood sample from an infected individual, smear it out on a glass slide that's been carefully cleaned, fix it with alcohol, and then stain it with a variety of reagents, Wright stain, Giemsa, and there are lots of other stains that have been used to reveal the parasite within the red cell. And all of malarial parasites begin their life cycle as a small merozoite that's penetrated inside the red cell, and this stage is known as the signet ring stage. It actually has the appearance of a signet ring. And every single one of these parasites, the Malariae, Vivax, Falciparum, Ovale, all of these parasites start their life in this fashion. What are the consequences of infection? Let's not worry about what happens to the mosquito. If they die, they die, but of course they don't die mostly. They survive. What happens to us? Well, the first thing, of course, is to acknowledge the fact that because it's a parasite of red cells, that the more red cells that are destroyed, the more likely it is that we will suffer from the lack of those red cells, and that condition is known as an anemia. And a simple red cell destruction process results in a simple anemia. It's not so simple, of course, but the fact is that it's not due to the lack of iron, it's not due to the lack of a nutrient, it's due to the lack of the red cells themselves. And the liberation of parasite material from its metabolism and the leftovers from the erythrocyte contents into the circulation induces a fever. So we have an anemia and a fever portion to the life cycle. And because we're reducing the number of red cells, we have a reduced capacity to absorb oxygen. And so just shortly after the release of these materials into they would be called pyogenic materials, into the circulation, the patient, first of all, gets a fever, followed by the fact that they cannot warm their bodies up enough because their oxygen carrying capacity has been reduced, and they start to shake with chills. So chills and fever are the main signs and symptoms of a serious infection with malaria. The other things, lots of other things happen, of course, and they're related to the fact that Plasmodium falciparum has this unique ability to sequester itself in the precapillary arterioles. And when it does so, it slows down the circulation. And so we have a lot of organ failure symptoms and signs related to the fact that this parasite can interfere with the flow of blood. And if this occurs in the brain, of course, then lack of oxygen to the brain can result in a reduced brain function. And indeed, one of the consequences of infection with Plasmodium falciparum is a coma-like condition. That coma can result in death, or patients can eventually recover. And it's been learned by studying patients that have received numerous infections over the time of their life that indeed this reduction in brain function during the acute phase of the infection has an effect on their ability to reason afterwards. And so we can see that even though people can survive this infection, let's say that they're young adults and they've never encountered it before, but they've had the advantage of growing up in an endemic area, so they've had maternal antibodies, they've been raised in an area where everybody has got this infection all the time. These people that get infected and recover and infected and recover are less able to carry out life than they would be if they hadn't been infected with regards to higher brain function. Here we see the reason, the molecular reason why this parasite is so successful in attaching itself to the endothelial cells. These are the histidine-rich knobs on the surface that the parasite engineers basically by living inside the cytoplasm, well, not quite inside the cytoplasm of the host, but somehow manages to get this histidine-rich protein across the membrane that it's living inside of and into the cytoplasm of the red cell, and it translocates just under the surface of the red cell membrane. And these are the points of attachment to the endothelial cells. And here's an experimental infection in an Aedes monkey to show you what the consequence of that is. This is a section of brain tissue that shows you a very heavy infection with Plasmodium falciparum. In order to get the Aedes monkey infected, they had to remove the spleen because in ordinary circumstances, the spleen of the Aedes monkey is its main line of protection against this infection, and therefore it doesn't become infected. Remove the spleen from an Aedes monkey, and it becomes as easy to infect as a human.